Hi students, today we are going to deal with another non-thermal processing method called ultrasonic processing. Ultrasound is the energy in the form of sound waves in the frequency range of above 20 kHz. Ultrasound is similar to normal sound in its physical properties except that it is not audible to humans. This limit varies from person to person and is approximately 20 kHz in an healthy young adults. The term sonic is applied to ultrasound waves of very high amplitudes. Many animals have the ability to hear sounds in the ultrasonic frequency range. Animals such as bats use ultrasound for locating prey and obstacles. Ultrasound has been used in many different fields for a variety of purposes. The first technological application of ultrasound was an attempt to detect submarines. Ultrasound devices are used to detect objects and measure distances. Ultrasound imaging or sonography is often used in medicine. Also used in the detection of flaws in concrete buildings, synthesis of fine chemicals, and in the treatment of diseases. Ultrasound make use of phenomena that are fundamentally different compared with those applied in conventional extraction, processing or food preservation techniques. Many food ingredients and food products are thermally sensitive and vulnerable to chemical, physical and microbiological changes. Due to these, losses of some components may occur. Also, low production efficiency, time and energy consuming procedures may be faced during the use of conventional food processing methods. These shortcomings have led to the dawn of a new sustainable, green and innovative techniques in processing and preservation of food, which typically involve less time, water and energy. One such technique is ultrasound assisted processing of foods. On completion of this module, you will be able to understand the basics of ultrasound and its mechanism of action in food processing, and also able to identify the different applications of ultrasound in food processing. Before going into the topic, you must know about ultrasound. Ultrasound, when propagated through a biological structure, induces compression and depression of the medium particles, and a high amount of energy can be imparted. The intensity and the frequency of sound used in a processing unit depend on the type of application. The application of ultrasound is broadly classified into two groups based on their power and frequency. They are classified into low intensity high frequency ultrasound and high intensity low frequency ultrasound. Low power high frequency ultrasound is normally used to monitor product or process, mainly for non-destructive analytical technique in food processing. It is also utilized to obtain information about the physico-chemical properties of food, such as composition, structure and physical state. High power low frequency ultrasound is normally used to alter the properties of a food material or to enhance the progress of a process. For example, modification and control of crystallization process, dispersion of aggregate materials, degassing of liquid foods, producing emulsions, enzyme and microbial inactivation, enhanced drying and filtration, and induction of oxidation reactions. Ultrasonic has been used in combination with other methods to get additional benefits of other processes. The combined techniques are shown to be effective in inactivating microorganisms and enzymes. Such combined methods are thermosonication, that is ultrasound in combination with heat, manosonication, that is ultrasound in combination with pressure. Manothermosonication is ultrasound in combination with both pressure and heat. Both thermosonication and manothermosonication are capable of reducing processing time considerably and have potential of energy efficiency. With regard to the application of ultrasound in food processing, ultrasound makes use of physical and chemical phenomena that are basically different from those applied in conventional extraction, processing and preservation techniques. The potential applications of high-intensity ultrasound in food processing are microbial and enzyme inactivation, extraction, drying, freezing, thawing, crystallization, filtration, demolding, cleaning, degassing or defoaming, cutting, mixing, emulsification and homogenization. Let us now see how ultrasound inactivates the microbes and enzymes. Though the conventional heating of foods aids in preservation, it also causes reduction in nutrients and organoleptic properties of food products. The lethal effect on microorganisms by ultrasound was first identified in seawater bacterium Bacillus fisheri. A reduction in light emission from the bacterium was observed when it was sonicated at 375 kHz under temperature controlled condition of above 19 degrees Celsius. Also reported that all the bacteria appeared dead when viewed under microscope, and also inferred that the microbial death was due to cell disruption caused by cavitation. The microbial inactivation during sonication may be due to three reasons: cavitation, microstreaming, formation of free radicals. On sonication of liquid products, the sound waves that propagate into the media and result in alternating compression or high pressure and rarefaction or low pressure cycles depending on the applied frequency. During the low pressure cycle, ultrasonic waves create small vacuum bubbles or voids in the liquid. When the bubbles attain a point at which they can no longer absorb energy, they tend to collapse violently during a high pressure cycle. This phenomenon is termed as cavitation. Cavitation is associated with shear disruption, localized heating and free radical formation. During this collapse, a very high temperature of approximately 5000 Kelvin and pressures of approximately 2000 atmospheric pressures are reached locally. Cavitation is considered as one of the main reasons for microbial inactivation. Also, microbial inactivation occurs due to the inability of microbe to withstand the extreme pressure variation taking place during the cavitation process. The second mechanism of microbial inactivation is due to the generation of microcurrent, or simply called as microstreaming. The microcurrents created around the bubbles has the ability that they can catalyze chemical reactions and disturb microorganisms. The collapse of bubble during cavitation results in liquid jets of up to velocity of about 280 meter per second. The third mechanism involved in microbial inactivation with ultrasound is the formation of free radicals and hydrogen peroxide, both of which have bactericidal properties. You know that enzymes are highly sensitive to thermal treatment. However, when the enzymes have higher thermal resistance, an elevated or higher temperature and a longer holding times are required for their inactivation. The exposure of food to elevated temperature for a prolonged period negatively affects the quality of food. The application of ultrasound considerably reduces the enzymatic activity. Cavitation and formation of free radicals can lead to an enzyme inactivation. At sufficiently low levels of sonication, changes in structure and metabolism can occur in cells without distracting it. The activity of peroxidase in fruits and vegetables are associated with the development of off flavors and browning pigments that can be reduced significantly by ultrasound. Thermoresistant enzymes such as lipase and protease can be inactivated more effectively by manothermosonication. Now we will see how ultrasonic waves helps to extract components from raw materials. Ultrasound has been used to extract numerous components such as oil, protein and bioactive components from different sources. Integration of ultrasound during extraction can accelerate heat and mass transfer. The ultrasound waves propagate through the material and alter its physical and chemical properties. The mechanism of action for the enhanced heat and mass transfer rate during extraction is due to the cavitation effect, which breaks the cell wall and facilitates the release of cell content into the extraction medium. It can also facilitate water uptake by the material during sonication, so cause an enlargement in the pores of cell wall. The mechanical effects of ultrasound provide a greater penetration of solvent into the cellular materials and improve the mass transfer rate. The method avoids the usage of enormous quantity of solvent and also reduces the time of extraction. A study shows that sonication accelerated sugar diffusion and yielded high level of dry matter content and sugar content in juice. Ultrasound extraction has been successfully carried out to obtain herbal extract from fennel, hops, marigold and mint, carnosic acid from rosemary, carvone and limonone from caraway seeds, ginsenosides and from ginseng, and anthraquinones from noni root, chitin from prawn shells, lutein from egg, polyphenols, polysaccharides, carotenoids, anthocyanins, tartaric acid and various aroma components from different type of raw materials. Next is the application of ultrasonic drying. Ultrasonic drying can be carried out at low temperature than conventional methods. This emergent and promising technology finds its application in drying foods by having vibration in contact with the food. This method reduces the probability of oxidation or degradation in the material, and drying occurs without affecting the essential quality characteristics of food products. By employing ultrasound, the heat transfer between a solid heated surface and liquid is increased by approximately 30 to 60%. Also, the effective water diffusivity increased after application of ultrasound during drying and also causes a reduction of about 60% in the drying time. This increase in moisture diffusivity is maybe due to the formation of microchannels during the application of ultrasound in the structure of the food material. Most commonly, the ultrasonic energy is combined with hotter drying or osmotic dehydration. This can also be used under the vacuum condition, which intensify the movement of water molecules and facilitate evaporation. It has been studied that ultrasonic energy coupled with hotter drying reduced the input power to approximately 20% in comparison with non-treated ultrasonic drying system. Also, it leads to a decrease of 6% in the final moisture level and 16% reduction in the drying time. The advantages of ultrasonic drying over conventional drying are enhanced energy efficiency, low temperature drying, better quality products, reduced environmental impact and safety of the system. Next application is filtration using ultrasound. Filtration is used to separate solids from liquids and is an important step to many industries. This separation can be either for the production of solids-free liquid or to isolate the solid from its mother liquor. Incorporation of ultrasound to filtration and screening causes the filter or screen to vibrate, creating a frictionless surface, allowing the liquid or smaller particles to pass through more rapidly. The common challenge faced during membrane filtration is the formation of fouling, which is due to the presence of natural organic material. The natural organic material in water usually blocks membrane pores, binding particles together to generate low permeability cake layer on the surface of membrane. The use of ultrasonic assisted membrane filtration drastically reduces the fouling and improves the overall performance of the membrane. Ultrasound assisted membrane filtration has been studied for products such as milk, whey protein, fruit juices, bovine serum albumin, baker's yeast, peptone, dextran and polyethylene glycol. Next, the use of ultrasound is during mixing, emulsification and homogenization of food ingredients. Ultrasonic bath or horn system is used so that ultrasonic vibrations are generated via the flow of a liquid. The resultant shock wave produced near the surface of two immiscible continuous and dispersed phases during cavitation facilitates the efficient mixing of two layers. Comparatively, low energy input can result in the formation of very fine, highly stable emulsions than conventional mixing. Ultrasonic mixing finds its application in food products such as fruit juices, mayonnaise, dough preparation and tomato ketchup, and also non-food applications in petrochemical, chemical, textile, cosmetics and pharmaceutical industries. Ultrasonic homogenizers helps to reduce the size of particles in a liquid to improve uniformity and stability. The reduction in the mean diameter of the particle increases the number of individual particles and thus leads to increase the particle surface area. In a study during ultrasonic homogenization of milk at 20 kHz, it was seen that the size of fat globules in milk sample was much smaller than the size of globules treated by conventional homogenization process. The reduction in the size of fat globules was attributed to the physical effects generated during acoustic cavitation. Also found that in ultrasonic homogenized milk, a significant improvement of water holding capacity and viscosity was observed. Similarly, reduced syneresis was noted in yogurt obtained from ultrasonic treated milk. For any manufacturers, cutting or slicing a portion of food product is always a challenge, especially to retain the appearance, quality and presentation of the finished product. Mechanical blades are commonly used in the food industries. These mechanical cutting blades find its limitations for cutting delicate and soft food products. The use of ultrasonic cutting offers considerable benefits in terms of yield, quality, output and appearance of the food products. The process is carried out by using high frequency oscillation of a sonotrode or cutting blade which easily and cleanly pass through the product being processed. The oscillation of the blade ranges from 20 to 35 kHz depending on the product. The high frequency blade reduces reactive cutting forces and the product is cut neatly without crushing or crumbling or smearing, which is generally seen in conventional mechanical cutting systems. Ultrasonic cutting or slicing has been successfully applied in products such as cakes, cheese, pizzas, meats, bread rolls, sandwiches and cereal bars. Degassing and defoaming can also be coupled with ultrasound. Ultrasonic degassing is used to remove entrained or entrapped gas bubbles from a variety of liquids including water, beverages, melted chocolates, oils and emulsions. The purpose is to displace the air from the liquid surface in order to avoid organoleptic damage of the product by bacteria and oxygen. The ultrasonic degassing technique decreases the number of broken bottles and overflow of the beverage when compared with conventional mechanical agitation. With regard to food industry, this technique is used to degas carbonated beverages before bottling. Ultrasonic degassing is rapid in aqueous product than the viscous liquids. The most common conventional method of defoaming is the use of chemical antifoaming agents. Though the chemical agents are found to be effective, they tend to contaminate the process. Based on the products, foam is sometimes considered as an unwanted product in the industrial process, which pose problems in process control and in equipment handling. For example, during fermentation, foam can result in the loss of culture fluid and cells, contamination of the environment, formation of an barrier to oxygen transfer and promotion of cell lysis. Hence, a distraction or control methods to reduce foam is necessary. The use of high intensity ultrasound waves is a clean method to defoam without having a direct contact with the product. You know that freezing is a means of preservation of foods by converting the water into ice. The crucial factor in freezing is the formation of ice crystals during the freezing of water present in the food material. The challenging part of freezing is the production of uniform ice crystals. Non-uniform ice crystals leads to damage of food structure and loss in sensory attributes, which is common in conventional freezing methods. Ultrasound assisted freezing facilitates rapid and uniform ice crystal formation. The formation of small ice crystal reduces the damage to the products. During ultrasound assisted freezing of dough, it is seen that ultrasound considerably increased the freezing rate during phase transition stage of freezing. It is also observed that it reduced the freezing time by 11% at an ultrasound power level of 288 watt and 360 watts. Another study on freezing of potato using ultrasound assisted freezing exhibited a superior cell structure compared to the conventional freezing. About the advantages of ultrasonication, ultrasonication offers numerous advantages in the food industries, some of which are: ultrasound waves are environmental friendly, non-toxic and safe to use in food systems. It is highly effective against microbes, spores and enzymes. Internal defects of products can also be detected using ultrasound. Ultrasound combined with other non-thermal method is effective against microbes. And this process requires low running cost and it is simple to operate and effective power output. It improves the yield and rate of extraction as compared to other conventional methods. This involves minimum loss in flavor and yields superior consistency of product. Regarding the disadvantages of ultrasonication, inactivation of the released product is caused due to shear stress developed by swirls from the shock waves. Depth of penetration of ultrasound is affected by the product nature. Generation of radicals as a result of critical temperature and pressure condition may cause some changes in food components. Ultrasound wave frequency can impose resistance to mass transfer and potential problem with scaling up plant in ultrasonication. Let me summarize the topic. Ultrasound waves are in the frequency range of above 20 kHz, that is above human audible range. Application of ultrasound may be low intensity ultrasound or high intensity ultrasound. Combined techniques of ultrasound with heat and pressure are proved to be effective against microbes. Numerous potential applications of ultrasound have been identified and also commercially implemented. Cavitation is the main principle behind ultrasonic inactivation of microbes and enzymes. Integration of ultrasound during extraction can accelerate heat and mass transfer. Ultrasound assisted freezing produces uniform ice crystals. Ultrasonic energy has the capability to produce very fine, highly stable emulsions than conventional mixing. Thank you.